So let me move on the next presentation. Professor Michael Strano. Professor Michael Strano is currently on the carbon dubs professor in the chemical engineering department at MIT. My professor, Strano, received the numerous awards, including the 2005 Presidential Early Career Award and also many other prestigious awards. Then Professor Strano is currently an editor for the journal Carbon and the Protocols in the Chemical Biology. And also the Professor Strano was elected to the National Academy of Engineering in 2017. Please welcome the Professor Strano.
Wonderful. Thank you very much. Thank you. And it's my pleasure to be here. I just want to confirm that my slides are presenting and that you can hear me.
Okay, perfect. Thank you.
Perfect. Okay, great, good. Well, it's my pleasure to be here. I want to thank the organizers. This has been a wonderful conference, and my only regret is that I'm not there in person to join my colleagues. I hope this Zoom talk will be a sufficient substitute. I wanted to highlight work today fitting within the session, emerging materials for the environment and for sensing. And so the title of my talk today is Nanosensor Coupling to Human and also Plant Interfaces for Real-Time Information Transfer. And I'll explain why we become interested in plants. I'm actually currently in Singapore now. The Singapore government actually funds a satellite laboratory that I use. It's called DISTEP, Disruptive and Sustainable Technology for Agricultural Precision. And that's where we do, among other things, sensors and plant technologies for agriculture. So I just want to start off by thanking my research group. They're the engine that makes everything happen. I was part of a program that helped us to produce a mission statement. We worked hard on it, so I want to relay it. It's in order to improve the human condition. The Strano Research Group seeks to understand nanometer scale phenomena using mathematics and chemistry, and also invent new technologies for health, energy, food production, and material science. And in fitting with the session today on sensing, I wanted to focus on two topics of our work, particularly, let me see if I can, particularly health and food production. I thought we would start off on our work on agriculture, and so because I'm the first speaker to highlight this promising area of nanotechnology, a growth area for nanotechnology, I thought I would give a little motivation as to why that's of interest. I'm a chemical engineer by training, and so at first it might be an odd fit to say why is this chemical engineering interested in agriculture? But I want to start off by convincing you that there's a global level problem that's facing humans. This is if you look at UN global population projections. So we've already exceeded 8 billion. I think the 8 billionth person was just born in the Philippines a couple weeks ago, and so this growth, which is exponential and saturating, but still growing at a considerable rate. If you consider population growth with what's happening to arable land on the next slide. So arable land for producing agriculture, this is where most of our agricultural products come from, the vast majority. Arable land is shrinking from a variety of reasons, mostly climate change. So by 2050, arable lands will shrink by 12 million hectares. And to put that into perspective, that's about the size of Belgium. And because I'm sitting here in Singapore, I can compare it to Singapore. It's about 160 Singapores. And this is going to have a dramatic impact on global food production. And so it's a technological problem, and I'd like to get my colleagues and all of the engineering talent we have around the world to try to address this challenge for humans. Both a challenge And an opportunity is that also by 2050, if current trends continue, and they're likely to, about 70% of the world's population will live in these mega cities. And the growth of mega cities brings its own technological challenges, but also an opportunity. Um, in that there is significant efficiencies in terms of transport and carbon footprint that are built into having us live close by. And so if you go into the grocery store, one of the things is I teach my children that when you look around at the foods in the grocery store, they have built into them a distance. So because of globalization, if you look at where our food comes from, food has really food. You could think of it as food miles, right? So some foodstuffs are locally sourced, but some may actually come and be delivered thousands of miles. And so each of those miles has an energy cost, has a carbon footprint. So we need to start to think about the supply chain, which is very efficient, but is also embedding these externalities that are affecting our planet. So the next agricultural revolution, which forward-thinking countries like Singapore, but also others around the world are interested in facilitating, is the next agricultural revolution. Agriculture, if you can't go outward, if we're shrinking land, then we have to go upward. So vertical farming in different forms, indoor farming. Um, uh, the idea here is if you run the numbers, you can collapse about 100 hectares. This is how agriculture is measured. About 100 hectares into one hectare, and the idea is that you benefit from internally from increasing production cycles. You can have many harvests per year, and that makes up for maybe an annual harvest from conventional farming. But I believe technologically we're not ready to really make this work. This revolution needs a new generation of engineering tools. So that's my pitch, and that's where we start. We were I'll start off with a little bit of background. So function starts with energy, and you might remember some plant biology from your high school or your college classes. So photosynthesis inside the chloroplast is where this carbon fixing reaction. That's probably one of the most important ones on the planet. Carbon dioxide and sunlight are fixed into saccharides, and that happens in the chloroplast. The chloroplast is not a plant cell; it's an organelle. And from a practical standpoint, as the start of our work, chloroplasts are actually pretty easy to extract from plant materials. I show an example here. You can take spinach, you can grind it with a mortar and pestle. You do what's called a Perkel separation, and you get these things that look like living cells, but actually they're really lipid. They're double lipid bilayer bags of proteins that continue to do this chemical reaction. You can look at them under the microscope. So our and we had noticed a couple of years ago there's been very little work on the chloroplast. A lot of work on photosynthetic organisms. Um, RCs are reaction centers, but very little work on the chloroplast. So we started studying chloroplast and particularly how they interact with nanomaterials. We found something very surprising. It really changed the course of the direction of my work. So here I show an isolated chloroplast in the video. Just want to make sure the video is showing. I've had trouble with this, so make sure. Okay. Uh, um, you have what you're seeing are fluorescent single-walled carbon nanotubes flowing past the chloroplast, but they're entering it at such high rates that they actually light it up. And so these particles are able to seamlessly inject themselves directly into this chloroplast, which by plant biology standards was very puzzling. And the chloroplast is not like a cell; it does not have the same kind of active mechanisms to bring the particles in. So the first question you might ask are they just absorbing to the outside? Well, you can do plenty of experiments. Here you show that's a single fluorescent carbon nanotube, and it's entering, and it's clearly bouncing around the inside. It shows the signature of confined diffusion. You can also do confocal. you can do a lot of experiments to show that the particles are entering the inside of the chloroplast, and they seem to be behaving in a kinetically trapped way. So it was our process of trying to understand this delivery into plants. We expanded outward, and this phenomena can even occur for certain nanoparticles. For some it works, and some it does not, and I'll explain why. We have a theory, but you can actually infuse a nanoparticle solution to the leaf lamina. It'll go into the vasculature, and you can even get this process to happen even in vivo. So what you're supposed to see here is that in the plant, if you look at the chloroplast, you can actually get you can actually find that you can deliver nanoparticles in the same way into the chloroplast. You see from the overlay, and we showed this in 2014, and the results were puzzling at the time. If you look for single-walled carbon nanotubes, you can actually show that it's highly dependent upon the wrapping. So if you wrap the carbon nanotube in DNA, that has a negatively charged backbone. The zeta potential is negative, and you see in fact that you get uptake into the chloroplast. If you wrap in polyvinyl alcohol, it's still negatively charged, but the zeta potential is not as big. You get no uptake at all. You cannot find particles inside. But it's not just the negative charge. If you wrap in chitosan has a positive zeta potential. You'll find uptake, not as much, but you definitely see it. And then a key signature here is that if you wrap in the lipids that make up the chloroplast, you actually won't get uptake at all. And so even though this is negatively charged, so it works for some corona positive or negatively charged. And this was a very exciting puzzle for us. In particular, you can take a look. It works for negative and for positive, but the magnitude of the zeta potential has to be large. This is not just for carbon nanotubes, but I show here are some of the results. It works also for the protoplast is the plant cell that you can extract, and you can see here. You see DNA wrapped carbon nanotubes going into the chloroplast. You see these organelles here, and also chitosan. And you see these for, but again, for polyvinyl alcohol, you do not see the uptake. And this works also for streptavidin conjugated quantum dots, and also di-conjugated gold nanoparticles. Here you see both have negatively charged zeta potentials, and both will actually go into. In this particular case, you see that streptavidin coated quantum dots will actually not go into the chloroplast, but they'll stay in the cytosol. And however, these gold and these gold nanoparticles as well you can find in the protoplast. So so the trend is that highly charged nanoparticles localize in the chloroplast, while more neutrally charged particles they're either localized in the cytosol or in many cases don't enter the plant cell itself. So we developed what's turned out to be a very powerful theory to describe this. And here's what we understand to be happening. Any charged nanoparticle, if it's in even a moderate ionic strength medium, it'll have a charge. It'll produce an ionic cloud around it. And then as it approaches the double lipid bilayer membrane, that ionic cloud is known to soften the membrane locally. So that this lipid softening has been described. There are equations for it. So this change in ionic strength will soften the lipid locally. And then what we propose that happens is that there's a lipid exchange to unsaturated sites on the nanoparticle. This lipid exchange draws the particle in, but then what appears on the other side is now a lipid-bound nanoparticle complex that lacks that driving force to go back through the membrane. So you have to explain why this process is not reversible. The particle then becomes kinetically trapped. For as long as it has these lipids, it can stay within the double lipid bilayer membrane. And you can put, let me say, you can put math in another kind of talk. I can I can go through the equations for step 1, step 2, step 3, step 4. You take all of the equations, you put them together. This is not the kind of talk for that, but you get a quadratic equation that you can then solve. And it's interesting that it's quadratic because it suggests this is the threshold zeta potential for a nanoparticle to enter a barrier inside the plant. And notice it's plus or minus, so you get two solutions. You get these blue curves for the chloroplast. And if you apply this just to nanoparticles across the field, here we have our gold. NC is nanoseria. You've got dextran. You've got chitosan. You see that all of the black points are found inside the chloroplast. The red are not. And you see that the equation does a very good job in plotting size versus zeta potential in predicting. It's a predictive. And it takes into this equation actually has essentially no fit parameters. It takes into account the size, the dielectric constant of the membranes, membrane mechanical properties, and this change in the enthalpy of absorption. This enthalpy change on lipid exchange is the driving force. And it works for both chloroplast and for the plant cell itself. So to explain that a little bit better, you've got this threshold zeta potential to bypass a particular membrane. For the chloroplast, it's all you have to change is the membrane dielectric constant, and you get these two curves, and it explains really well the particles that are in the chloroplast and all the particles that are not. If you change this value to 7.8 to now reflect this single lipid bilayer membrane, now actually you can predict particles that enter the plant cell, but don't go into the plastid. And so you get this equation that's extremely predictive. It can basically allow us to design a particle. It gives us really unprecedented control. We can make a particle, and we can control whether it'll flow in the vasculature of a plant, enter the plant cells, but stay in the cytosol, or enter the double lipid bilayer membrane. And to the best that we can tell, this equation applies across plant species, and it applies across nanoparticles. We developed this theory originally just to describe the carbon nanotube results, but we ended up with an equation that seems to be fairly broad, and it's also surprising that a deterministic equation like this can have such high fidelity for something as complex as these living organisms, these living plants. So as we started to talk about this with our agricultural colleagues and give talks about this, one question that kept coming up is, well, can you help us engineer new plants by delivering a plasmid to help help interface with the biomanufacturing machinery inside the chloroplast? For example, I should say the chloroplast is an organelle. It has its own genome, so it synthesizes distinct proteins there, and that's where photosynthesis occurs. And the chloroplast and its photosynthetic machinery have really been off limits or really difficult to access by conventional molecular biology techniques because it's been very hard to get things into the chloroplast. So we developed a series of nanocarriers using this LEAP, and I'll show you. We published this. LEAP is very, very predictive. So here, this is a chitosan nanotube carrier, and if you change the ratio of plasmid DNA, which is negatively charged, and the chitosan, which is positive, if you change the ratio, the net positive charge on the complex increases. And if you take a look at 1-to-1, your charge is such that LEAP predicts that you don't go into the you won't go into the chloroplast. And in fact, that's what we see here. You see in this middle, you see that a 1-to-1 does not produce any yellow fluorescent protein. And then if you go to a 1-to-6 ratio, your zeta potential now goes up to 35, and we see yellow fluorescent protein expression. So this LEAP gives us some more precision than we really deserve. It's a predictive model, and it can allow us. It allows us to make particles that can cross this biological boundary with some prediction. And we can actually do this in arugula, and so you can see this. You see the yellow fluorescent protein. This previous work was with extracted chloroplast in a protoplast system, so that's the extracted plant cell. But this is actually in mature arugula, and so you can see that if you look at in the tissue, you can get this expression to occur in the living plant. So here's the 1:6, and then reviewers asked us to do as many species as we could. We did arugula, we did watercress, tobacco, and spinach. In particular, it's noteworthy. There's a lot of molecular biology that's done with model plants like tobacco. It's easy to work with. But these other plants, these crop important plants like spinach, watercress, and arugula. At this point in Singapore, we've done more than a dozen crops. The techniques work very, very well. And so you're supposed to see here that these tools and LEAP appear to be species agnostic. Once we develop a technology, it seems to work in a variety of species. So we're not just interested in delivering plasmids. Coming back to the focus of today's session is really sensing. So we've been interested also in turning plants into sensors, and then later I'll show you sensing inside of plants. So this is work that we did in 2017, where we started asking, can a living plant be an environmental sensor? Can a living plant itself operate as a sensor? Plants have a big advantage there. Plants, they naturally quote unquote pump water from the soil up into the leaves, where they can act as a pre-concentrator. I'm in a sensing session, so I can say that plants do a natural pre-concentration of semi-volatile or non-volatile species into the mesophyll. So they're sort of a built-in self-powered sensor with or self-powered microfluidic conduit. They pump themselves with transpiration from the sun. They pre-concentrate. If you put here, I'm putting in a sensor for nitroaromatics, an explosive sensor based on fluorescent nanoparticles and a reference. So you're seeing here two spots, a fluorescence and a reference. And what we show is that you can actually, if you spike the soil at some time t = 0 with picric acid is our model explosive, you can actually turn the plant into a living groundwater sensor for this explosive here. So hopefully this movie will play. So this is a movie. So this shows you a little bit of the instrumentation. That's a living spinach seedling, and its roots are wrapped in cheesecloth. It's a common technique. We're putting water with a trace amount of picric acid here. You can see it has the discoloration, but you can do all this detection just from a standoff distance. This is a Raspberry Pi camera. It's basically a cell phone camera, and you see here that you get you get an infrared modulation on the right, but we have an infrared reference on the left in the plant. You can get this information. You can get it to transduce and be sent to a user's cell phone. So this was a very important step in the technology, showing that you can put all these pieces together. With this demonstration, we actually then turned our attention to turning those sensors inward to detect, to make new tools for agriculture. And the Singapore government has supported us for the past five years to create this academic and technological center called DISTAP, Disruptive and Sustainable Technology for Agricultural Precision. It's an integrated research group. It's part of MIT's SMART program. I take responsibility for the name. It's this word salad name. It's my fault. But what DISTAP does is it takes these technologies, it turns them inward to try to intercept plant signals. And that has really opened up new avenues for exploration. So to motivate that, you can come to visit us at distap.mit.edu. Basically, the idea is that plants themselves collect an enormous amount of information. Light intensity, temperature, they're exquisite sensors for water. They know the first early signs when a pathogen has interacted with them. Insects, mechanical injury. The plants have this information. Humans do not have this information. So plants collect this information early. It's really been off limits. It's been a sensing problem to get that information that the plants have and act on in minutes. They act on it in real time and get that into the realm of humans. And so we sought out to apply nanotechnology to really speed up this closed loop to get these signals early so that we can intervene in the farm to increase yield. So I'm going to introduce a technology that we've been working on over the past decade. It builds on some of the discussion in this session. Is how do you recognize molecules with lock and key? You can use aptamers, you can use antibodies. This technique we developed at MIT is enormously powerful. I'm actually convinced there's not a single type of molecule that we can't detect with it. It's called corona phase molecular recognition. I'll explain it. We have dozens of papers on it. For a sensing nanoparticle, like a model particle is the single-walled carbon nanotube. It fluoresces in the near infrared. You can. It's typical that you wrap nanoparticles in a coating to get them to be dispersed into a solution. So we call that a corona phase. So I'm supposed to show here these absorbed molecules. They help the particle to interface with the solvent. But that corona phase also completely controls the molecular interaction between the particle and an analyte. And so we recognize that corona phases can be pinned in place on the surface of a nanoparticle and adopt a three-dimensional configuration, much like if you think about the way a human antibody works. The intermolecular forces, the intramolecular forces of the peptide, say, they fold it into a secondary and tertiary structure that then form a binding pocket. And in much the same way, these phases on the surface of a carbon nanotube or a nanoparticle in general can enable recognition. So when it does, we call this corona phase molecular recognition. And it's a technique. I can give a whole seminar on it, but basically we make a library of these polymers. And we have a lot of design information that we can put into the polymers. But at the end of the day, we try to get a library just large enough that we can screen. And the technique actually is so useful now. It actually it seems to have enabled molecular recognition of almost any type of molecule that we've had access to. So some advantages of using a carbon nanotube sensor is that for the purposes of this talk, is that they're non-photo bleaching. So when we put them into the plant, they won't photo bleach over time. They can absorb and fluoresce in regions outside of where chlorophyll would form some interference, and they're largely plant biocompatible. They don't cause a stress response inside the plant, and you can find windows where you can study optically phenomena inside the plant with the sensor itself not interfering. So for today's talk, I'm just going to highlight one of the sensors that we're using in this step, and it's illustrative. So if you wrap a carbon nanotube in this GT15 DNA sequence, you'll make an exquisite near-infrared fluorescent sensor for hydrogen peroxide. So if you add hydrogen peroxide here, you're supposed to see there's a fluorescent spectrum. You see that you get a modulation. If you wrap in this AT15 sequence, we use that as an infrared control. You see a bit of a modulation, but you're able to subtract for it. You're able to ratio these two, and you get what's called a ratiometric sensor. And we've used this to actually try to intercept the earliest stress signaling in a plant. So when a plant undergoes stress that will ultimately reduce agricultural yield, the first thing it does, it seems, is it produces a hydrogen peroxide waves. We think it's one of the most upstream chemical signals inside the plant. It happens within minutes. And if you take a look, what happens is that at least a few of these signals originate in the chloroplast. They will generate reactive oxygen species, and then through membrane proteins, they'll turn those into hydrogen peroxide. But it's a very special kind of a wave that's produced. The hydrogen peroxide then travels to a neighboring cell where it triggers more hydrogen peroxide, and so on and so forth, to make this chemical wave that moves much faster than diffusion. And so it's a soliton wave. We've done a lot of mathematics in the group about how these soliton waves travel. And if you take a look, the experimental setup is quite interesting. You can put a nanoparticle sensor inside the living plant, and you can detect basically free space. You can detect using laser excitation. This can be a meter away, but if you can detect, those with optics experience know, if you can detect over a meter, with today's technology, you can detect really from satellite. And so here's an expensive detector, but I'll show you data with that $20 Raspberry Pi as well. But that's really what's happening. We're really doing an infrared detection through the living plant. And what you see here is if you poke the plant, that's a standard for wounding stress. You see, similar to the experimental, explosive sensing, you're supposed to see here on the left is the hydrogen peroxide sensor. It's relative to, on the right, this AT reference, and you see here that you get a quenching that then slowly restores, and you're getting that within minutes. So you might think of your plant as sitting there static, but you're seeing in real time. You're seeing a chemical signaling response of the living plant. And you see that a little bit here. You see that it starts off here. You see the quenching occur, maximum of about 4 minutes, and then by 20 minutes it's completely restored. So this is the first collection of this information very, very quickly, and we can put it onto a user's cell phone. It turns out that this characteristic shape, we've seen it over and over and over again. We see it across different plants. It is the characteristic response of wounding, and this shape you can show is actually described by a logistic function, and it's here we show you can even detect it with this cell phone type of camera, and this wave is very robust. So if an insect bites the plant at 1 and then spot 2 and then spot 3, you'll see these waves pass through, and they can be picked up by the sensor, and they can be transduced to a user's cell phone. So you might think of a plant as being quiescent or static, or processes taking a very long time, and that's true. Gene expression can take a long time, but there is this immune signaling that happens within minutes. We're very excited to be able to detect it. So we've done a lot of mathematics on this chemical wave. This wave appears in a wide variety of crop important species like spinach, strawberry bile, lettuce. It's widely intercepted. Now, what happens is in different species, the characteristics of the wave, the size, the full width at half max, the lag, the amplitude, these are all modulated. They're distinct, but it's all this logistic function. So we've really found an evolutionarily conserved plant communication mechanism that we can now use to see some of the agricultural implications. I mean, you can a single insect on a single plant, the moment they bite the plant, you can get that information within a minute. It's an exquisitely sensitive detector of what's happening inside the plant. One of the most exciting things is that this hydrogen peroxide wave, in a multiplexing that we're still trying to work out inside the living plant, is that if you wound the plant, you make this waveform. If you do a conventional heat stress, there's a very conventional heat stress assay that's used in plants. It's a hot wire. Our hydrogen peroxide sensor also detects a chemical wave, but you can see that the wave form is distinct. It's very distinct from this red wave. Light stress also produces, again, a very distinct profile, and then pathogen also quite distinct. So this work introduces the idea of not just a signaling wave, but of a wave form. And so that's what we're introducing into agriculture is the idea that you can use this upstream wave form and you can distinguish the types of stresses that the plant is undergoing, and you get that with orders of magnitude faster than we ever did, and fast enough to where we can intervene and save the crop and improve yield. So with that, I'm actually going to switch gears to the last part of my talk. I think I have a few minutes. I'm going to switch from plants actually to humans. We saw a lot of fascinating technology to measure signals inside the human body or at the surface of the human body. I've been working on at MIT for more than 10 years this problem of how do we put optical nanosensors in plants? It's easy. A leaf is actually very, very thin, right? And but if you think about one of the most challenging spots to put a nanosensor, optical fluorescent, say nanosensor, would be inside the brain, deep inside the human brain. And so I want to talk about a big advance that we've made that allows us to implant nanosensors into thick tissue. And we've really solved the problem of how can you get an optical signal through thick tissue to be able to communicate optically through the human body. So you don't have to have wires. And that allows us, our goal is to try to access signals that are otherwise not possible. And this problem we have thought about for a while. It's a very difficult one. But my hope is to try to just like we're seeing in plants, these chemical waveforms. My goal is to try to see if we can detect signaling waveforms in the human body. Okay, to describe why this problem is so hard. First, humans are not transparent, and we're not thin like plant leaves. An optical signal has to travel through tissue, and there's strong absorption. So your blood has a red color, but its color is different when it's not oxygenated. But otherwise, it's strongly pigmented. So you can't get visible light to any appreciable amount through human blood. It's the primary pigment in the human body. Melanin also has a strong absorption. It's by design. But if you move to the infrared, you also have water. Water also absorbs infrared light. So this adsorption you have to overcome. You also have to overcome scattering. So in addition to absorption, light through the medium has to scatter, and scattering is very punishing. The shorter the wavelength, the scattering cross section goes up by a factor by a power to 4 or 5, or sorry, 3 or 4, and so it's very punishing. You can move to the infrared to try to mitigate it, but it's still strongly attenuating your signal. And there's also autofluorescence. So biomaterials, materials in organs and so forth, they constitute fluorescent materials themselves. So while you're trying to signal to a nanosensor, they are producing a background of material. And so I'll get right to our innovation. We thought about this problem for a while, and we just published WIFF is wavelength-induced frequency filtering, and it is an absolute game changer of a technique that enables optical sensors now to be implanted at really insanely deep distances inside of what we would consider completely opaque media. Okay, and so WIFF is called wavelength-induced frequency filtering, and it addresses this problem of how do I get a nanosensor fluorescent signal from deep inside a strongly scattering medium. And I'll explain the techniques a little complex, but I'll explain it as simply as I can. So suppose you have an optical nanosensor in red. Okay, so that's its absorption cross section. Okay, if you excite it on this peak, you'll get a fluorescent signal out. But it's embedded in a background. So the strongly scattering, absorbing, autofluorescing background, we'll say, is this black. Okay, so the total medium is a composite of the two, which I don't show. So if I excite the nanosensor, but I modulate not its intensity, but its its excitation. If I modulate the excitation wavelength at frequency f, okay, what'll happen is the component associated with the background, if you think of this excitation going this way, the component of the background um, will um, will be modulated at a frequency f, just the input frequency. But the fluorescent signal, if my center line is at the peak maximum of the nanosensor, this signal will be modulated at frequency 2f. So this technique allows you to not only self-reference to the background signal. I can ratio these, and I don't need another reference like I showed for plants. But I can also subtract out the background noise, and that's why it's a game changer. So to show this a little better here, in the simplest implementation, you can just do 3 different lasers and make this sort of heavy side type of sign. But as you excite the sensor and you excite the background, you'll get signals that are separated in frequency. So you can see here, your sensor frequency will be 2f, your background will be 1f, and you can separate them. And like I said, we implemented this with a 3 laser technique here. This is close to my last slide here. So if you take an FFT of the signal, you can clearly see just from the signal. This is the tissue background. This is the embedded sensor. You get 2 FFT peaks. You can ratio them. You can subtract them. It's completely. The ratioing also means that as a living animal or human, as they're moving, you can easily correct for that in a self-referencing way. And if I'll just show you here that what you can do here. This as an example, we put two sensors in, a hydrogen peroxide sensor and an ascorbic acid sensor, 2.3 centimeters implanted into probably the most complex tissue that you can think about. We've done brain. This is actually interperitoneal. You get a huge signal to noise improvement. It's not just an improvement, but you can actually get a signal, and you can do sensor detection inside this medium. If you don't use WIFF, there's really no signal above the noise that you can appreciably detect. And so this WIFF is a very powerful tool that we think will enable a new generation of sensors to go inside the medium, inside these complex media, and open up this space for biomedical sensing as well as agriculture and others. So with that, I think I'm out of time. I would like to thank my research group, and it's really the work that I showed is really just half of the research group. And I need to thank funding from the MIT SMART program, the Singapore government, and NRF. Our center is called DISTAP. And I talked about that. I'd like to thank the other funding agencies, and I'd like to thank the audience for your attention. And I'm happy to answer any questions.
Thank you very much for the excellent presentation, Professor Strano. Due to limited time, then I will take the 2 questions. The first question: how large biomolecule can be delivered or bound in the single nanotube while entering chloroplast? Short peptide can be bound on the singular nanotube for more specific binding?
That's a good question. That question comes up. The question is how large of a particle can actually go into the compartment. This goes back to the LEAP model. And the reason why that's important is because some users will have plasmids that are extremely large. So we get asked that question a lot. LEAP actually, if we go back into the slide, LEAP predicts a saturation. The LEAP mechanism predicts that above a certain size, there should be a threshold in intensity here. So if we look at the smallest dimension, above a certain size, maybe 40 nanometers, LEAP predicts that it doesn't matter anymore. But actually what happens is as the particle gets larger and larger, there are other considerations and forces. As you move out of becoming a nanoparticle to really a submicron particle, there are new forces that have to be considered for the work required to enter. It becomes non-linearly more difficult to open this up. In fact, if if something protrudes into a cell that's too large, it'll actually rupture and lyse the cell. So right now with LEAP, conservatively 30 to 40 nanometers, the mechanism seems to work very, very well. What that means in terms of a plasmid, you can back outward. We make these complexes, we put the plasmid on, and then we do measurements of the hydrodynamic radius before we put the material into before we put them into cells. So it's a great question, and more work needs to be done on how to get larger plasmids into the cell.
Thank you. And the last question. Thank you for wonderful talk. Is there any approach to maximize the number of nanocarrier inside a plant? And how about the thickness or size dependence on plants?
Yeah, I have a student working on that now. So LEAP is basically a thermodynamic theory. It predicts whether a particle has the energetics to go across the membrane. The question is, what about the expression levels? So that's the number of particles in a given time. And I have a student working on that. It's a very different kind of model. It's a kinetic model. And so we're collecting data. It's a lot harder. So just from my colleagues know, thermodynamics is very mathematical. You can and logical. And if you're willing to wait to equilibrium, you can analyze things easily. Kinetics much harder. And but it's an important question because it influences the expression levels. And so my answer, I'm going to punt on that and say that we're working on it, and it's a hard problem.
Okay. All right. So I would like to thank the Professor Michael Strano again for impressive presentation.